Okay, we finally have made our way through the transcription process as well as the translation process, and we're finally down to proteins. What actually are proteins? And you can remember that we said during the translation process there were a series of steps, and at the end of those steps, after termination, what you had was a string of amino acids, right? That's the end product of translation itself. You have not yet produced a protein. And it's the basic idea that you have a gene, the gene produces mRNA, which can be then matured and then read through the process of translation to make a polypeptide. And that's where we are now. We have made a polypeptide, this linking together of these units called amino acids. And we are going to now work toward making those into what we would think of as functional proteins. Okay. So obviously, the way that a protein will work, the way it's going to look, the way it's going to function, is going to be directly related to a group of amino acids that it has, and then any side chains, other things that can be connected to it. Now, before we really look at any of that sort of what we will call secondary structure, tertiary structure of these, we really need to think about how these amino acids, what they are, and how they're interconnected. This is just very brief. I know you've seen this other places, and certainly if you haven't, when you go to chemistry, biochemistry, organic, you'll see a lot of this information. But the basic idea for us is that we have these amino acids. They're very regular structure, right? There's a couple of carbons in the center and nitrogen in. The nitrogen in is called the amine in, amino. Okay, that's where we get amino acid, right? And then the other one is a carboxyl group that's found on the other end. And then there are always these R groups off to the side. We'll talk more about those, but these R groups are what define the amino acids. Again, I said before in other lectures, we have 20 common amino acids. That's why we have that coding dictionary, the way it's set up. And these R groups can be as simple as a hydrogen all the way to a complex ring structure. And of course, in a laboratory, you can attach anything here just about, so you can make very complex amino acids. But in the real world, in the functional world of living organisms, there's only 20 common amino acids. All right, well, these amino acids, but we cook those together into what we call peptides. So we need to talk a little bit about what a peptide bond is, okay? Peptide bond is a result of combining two of these amino acids from the one of the carboxyl ends to the amino end of the other. Okay, so we have our amine end, we have this carboxyl end. Remember that we had during the process of translation, we had this structures called that came into the large ribosome and helped us link together these amino acids, right? And we talked about a complex that we called Peptidyl Transferase. It's an enzymatic protein complex that helps to catalyze this peptide bond between these two areas. And it very simply does that by allowing the formation of this bond directly between the carbon and the nitrogen. Of course, there's some oxygens and hydrogens here that you could knock off. And in fact, you do. You end up knocking off two of the hydrogens and one of the oxygens. This is one of the places that we get metabolic water. Right? It's the production of proteins. Okay? So this is occurring all along throughout the entire elongation process. Each time a new tRNA would come in, you'd form a peptide bond, you would release off of the P site, and you would have this growing peptide, and there'd be release of cellular water in the process. So here's our peptide bond. Okay? It's important little bonds, and it was just continuing. We're going to continue along. We'll see a string of these in just a moment. Just as a note, the earliest antibiotics that was actually commercially available was one that was back in 1947, was developed to actually prevent this peptidyl transferase from operating correctly with things like with bacterial invasions. So it was an antibiotic used for that. Still commonly used now. It's used for originally for typhoid and other things. It's still used very commonly as sort of a backup antibiotic. It's used in veterinary medicine in really weird ways. I think one of them is koalas, little koala bears, right? They can get an infection like chlamydia type infection, and it's used to treat that. It's also become very important, or at least potentially important, frogs, right? We've lost almost, I forgot how many different species, but something like 20 to 30% of the frog species amphibians have disappeared due to this fungus that's moved up from Australia into places. But it turns out that this drug can actually treat that fungus. The problem is you can't, you have to put it on externally on the animals, and there's no way, we don't know what the secondary effect of doing that. You certainly can't treat broadly with it, but it has an interesting outcome that it's potentially useful to protect some of these amphibians. Anyway, it just breaks down this prevents this formation of this peptide bond is basically what it does by interfering with peptidyl transferase in these bacterial systems. We have a full length code here. Okay, so we're going to have our amino N over terminus. You always have an amino terminus, and you already have a carboxyl terminus. We always talk about the N and the C terminals, and those are really important because proteins, when they move through membranes and things, they'll have the C terminus out or they'll have the N terminus out, and there's actually really interesting ways that you can come in, and when you're trying to figure that out, like how are they oriented? Let's say maybe the mitochondria has a double membrane, and is the protein that goes through those membranes going like this so that the C terminus is out here and it's in, or is it like this? It turns out there are these chemical reagents you can put in, and you can knock off one or the other, so you can come back and look at what you sheared off of the protein. But anyway, it's a full-length polypeptide, so however many codons you have, each has a unique amino acid that's put in place along this polypeptide. And of course, all this relates directly back to the message. There was the cap. We talked about the fact that there was a non-coding region where we start putting them together, the whole process of translation, and you bring in a small subunit until you finally hit a real start code, which puts down methionine. Then you have what's known as the coding sequence, right? The actual part where the codons are read. Ultimately, there's a stop codon. There'll be a region past that where you don't translate anymore. And of course, the poly-A tail region, which has multiple functions. So this directly relates to that. Now, as I'll point out, many of these proteins, as we move through this, actually get MET sheared off and they get other things added to them. But MET is the first amino acid to go down, even though as a mature protein, a lot of times MET's been removed. Now, just to kind of give you an idea here of what we're looking at, these are all the side chains that make these amino acids form, and they can go all the way from simply having very simple forms like this to having these ring structure type things. So they can be quite complex. And unfortunately for you, if you take biochemistry, you will learn each one of these in detail and certainly much more appropriate ways of illustrating them in this. For our purposes, they're really, these three groups are most important for us as in talking about proteins. There's polar, there's non-polar, and then there's electrically charged. And these interact, and how and one of the really important ways that we're going to make proteins, okay, we're going to keep talking over and over about shape drives function of proteins, and we'll just hammer that home. The basic idea is that the way the protein folds and shapes itself, this polypeptide into this functional protein, makes it function, right? And it is often these side groups, often these side chains that interact with each other or do something, add on to other groups. They may interact directly with other amino acids, or they may interact with other polypeptides or other chemical elements in the cell to make that functional protein. Okay? But let's look at each of these and talk briefly about them, okay? These 20 amino acids, the side chains, and again, they have these tendencies to interact with each other in various ways. All right, so first are the non-polar. These are the hydrophobic. By definition, they tend to repel water, packed closely to each other. Okay, so these are where the amino acids would be. In fact, we talked about this occurring when we did histones, just dragging you way back. And we talked about the fact that histone was this octamer of proteins. These, the nucleosome, sorry, was an octamer of proteins that were histones. And the inner histones, and at least the areas that are in the interior part, tend to be hydrophobic, and they tend to be away from the water. The outer surfaces of those tend to be hydrophilic, and it's true in all proteins that these tend to be the ones where you see these hydrogen bonds forming, which allows the connection between the actual amino acids themselves. And of course, they're able to interact with water, and because they're on the surface, they often interact with other proteins and other molecules. And then the charged ones are the really important ones as well. These are very often on the outside. Remember that when we talked about nucleosomes again, we talked about those histones that made it up. And we said, oh, the outer surface of a histone is positively charged until it's modified. Well, that helps attract this negatively charged DNA, right? DNA is an acid, so it's negatively charged. So obviously, if we were going to look on the outer surface of these histones, these proteins that make up the nucleosome, we would expect to see some of these positively charged. And sure enough, histidine is very commonly there. Lysine is very commonly found on that part of the outer surface, not the whole protein, but on the parts that become the outer surface of the protein. So these are all very important functions, and they function in different ways to make that protein form and work. Okay? Now, given all this, all proteins carry some kind of charge in physiological conditions and pHs that you see in cells. And it's due to the fact that you've got some of these positive and negatives, okay? So you may have a series of amino acids like these blank ones, which have no charge, right? And then if you have some that have positive charge or negative charge, you can kind of add them up. And this little demonstration polypeptide that I have here has 1, 2, 3 positive charges and 2 negative charges. So it has overall net positive charge. This turned out to be really useful and important because we could do what was known as electrophoresis with this, right? Remember electrophoresis when we did, which you've already done in lab and places where we have those big gels and we run DNA through them, fragments. Well, long before we could actually do that effectively with DNA, we could do it with proteins. It was known as protein electrophoresis. It was a lot simpler than some of the sequencing because what you would actually do is you would grind up a piece of liver or muscle or whatever tree, a leaf or something, wherever you had these proteins that you wanted, you would put an electrical charge through there and they would move through these gels. These gels are often made of potato starch, so they're actually very safe. At the end, then you would actually pour on the ingredients you needed because many of these proteins were enzymatic, actually cause them to do something, to cause them to go into function as they would in the cell. And in doing so, they would release hydrogens or they would release some other area that could be captured. And once you captured it, you could actually get stains out of it. You could stain it and see it on the gel. So point of this is one of the earliest forms of genotyping that was actually done with proteins, not with the actual DNA that we now use more commonly today. And these are some gels that would reflect mutations that occur in some individual that form that had some kind of variation in the amino acids that were available. So if you say took away this positive charge right here and ran that against something that did have that particular positive charge, you get a different banding pattern and you can actually genotype those individuals. So it was interesting. Still used commonly today to look at cell variation within cells, etc. Don't use potato starch much, but there are other ways to do this, more fancy ways. But it's all electrophoresis. So that whole concept of electrophoresis, not just separating out DNA, it's actually much more useful than that. We can do all sorts of things. And in fact, doing two-dimensional electrophoresis, where you run it one direction and then take the gel, literally turn it 90 degrees and run it again, is the way that we would actually determine the amino acid sequence. It's called amino acid sequencing or protein sequencing. And it's done by doing a two-dimensional electrophoresis gel. One of the ways that we can figure out what these amino acid patterns are. All right, that's a general overview of proteins. Now we want to come back and say, okay, we know how we link them together. We know what a peptide bond is. We know what a polypeptide is. How do we get a protein? What's the next step? And that's what we're going to talk about next. What's going to happen to make those polypeptides into functional proteins?